idea here is to deprotonate this isoxazole ring, and that leads to ring opening, breaking of the NO bond, and that can be facilitated by a hydrogen bonding group coordinating to that oxygen. Here is a picture of a quantum mechanically computed thiozyme, which has, as I mentioned already, it has an acetate and imidazole. Here's the molecule undergoing reaction, and here's just a methanol to represent a serine or tyrosine or some other hydrogen bonding group. We then take this thiozyme and various proteins which are in the library. These are all proteins for which crystal structures are known, so we're not trying to design proteins from scratch, but take stable ones, but design new active sites. So we then look for matches in the active site region, and then when we find such matches, these are then redesigned by incorporating other side chains to get good packing of the transition state, and then these are evaluated. And let me tell you about some of the evaluations. Here's an example of a full protein with a new active site in it for this particular reaction. We evaluate this with molecular dynamics, so we use amber, which is a force field that's common for protein simulations. We put the protein in a water box, and then after equilibrations, we do 20 nanoseconds of molecular dynamics. So that is, all of the atoms of the protein and the water molecules around are simulated by molecular dynamics. And we do this for 20 nanoseconds, which we found is long enough to evaluate whether the active site design is maintained or not, or whether other things might happen. For example, we look for water accessibility, for the deviation of ideal distances from those that are in the designs and so forth. And here's just a evaluation, far too much data, but I just want to point out that these molecular dynamics do seem to be capable of differentiating active designs from inactive, at least of increasing the odds we'll get active designs. In green here shows some experimental data, catalytic antibody and a number of different enzyme designs from the Baker group and from Steve Mayo's group at Caltech, and these have all been expressed and isolated and studied as catalysts. These are the free energies of activation. These are the K-cats for these different molecules. And here are a number of designs that were inactive. The molecular dynamics, which were done some after the fact, some before the fact, but all of these were, what we found were that in these cases the catalytic contexts were maintained. This one we looked from dynamics like it probably would not be a catalyst. These had various bad things happen during dynamics that seemed to make them not candidates for catalysis. So the scorecard is pretty good. The green checks mean that we correctly reproduced the fact that there is catalytic activity. This one we got wrong. And of the inactive ones, these red checks were correct. The X's were wrong. So in any case, the dynamics does do a reasonable job of evaluating whether these designs are going to be good catalysts or not. Then, once we get through that stage and decide that something is going to be a good catalyst, we then do the experiments. This actually is proposing the way this will be done at UCLA, but it's done now in the Washington group or at Caltech by Mayo's group. So we order the genes, they get expressed in E. coli, and then the proteins are isolated and purified and tested for catalysis. And here are some results for this particular reaction. 2 different kinds of active sites were studied, with just a carboxylate doing the deprotonation or a catalytic diad doing the deprotonation. In some cases, crystal structures of these designed proteins have been obtained, and here's an overlay of the design in blue of the active site of one of the Kemp eliminases, as we call them, and in white, the crystal structure. And here are the statistics. 59 designs were made in 17 different scaffolds. Of those, only 12 displayed activity. The most active of those had K-cat over K-uncat of about 10 to the 5th. That was improved somewhat by directed evolution to 10 to the 6th. And Baker and Taufik in Israel are continuing to try to improve that. This can be compared with a catalytic antibody from Don Hilbert's group, which had 10 to the 4th acceleration. Let me conclude then by telling you two other successful stories. One is this retroaldol reaction. It's a substrate which was originally studied by Carlos Barbas, who developed a catalytic antibody for this, or several for this reaction. Here are several thiozymes that were developed for this retroaldol reaction. What we need to do is to form an iminium and then deprotonate this hydroxyl. So this is actually a picture of the iminium, which has been formed just, it would be from a lysine, but here it's just a methylamine. And then these other groups are present in order to deprotonate the hydroxyl group and to transfer the protons between the iminium and the hydroxyl region. Here's a different one. I won't take the time to go through it, but it's a different kind of active site design. Here again is a crystal structure, an overlay of the crystal structure with the design. So at least these design procedures do lead to proteins with the predicted structure. In this particular case, 72 designs were made in 10 scaffolds. 32 of these accelerated by 100-fold or more. The most active of these is 2.4 times 10 to the 4th. By comparison, catalytic antibodies are better in this case. So there's both one 10 to the 5th, another 10 to the 8th. Both of these are from Carlos Barbas, who was part of the enzyme design project, but also had previously done the catalytic antibody work. And you'll hear from Carlos tomorrow on a different subject. The end of the story here, we've successfully designed and synthesized catalysts for these three types of reactions. And of course, in the context of this meeting, our next challenge is to try to design and synthesize enzymes that might remove protein ages from modified proteins. This is the group that has worked on this at UCLA. We've been funded by DARPA and the NIH most recently. This is Gert Kiss, who had most to do with this talk that I've just given. And as I mentioned, he'll be giving a poster later this evening. Thank you very much.